worked in olfaction. She has worked in photoreceptor axon guidance, it is wonderful to have someone who is working in insect systems. Sorry, my mistake. But in organisms that have to have three-dimensional structure and differentiation, and where space, time, and cell interaction become extremely important. And she's going to be describing how to develop circuits to probe development in Drosophila. Thank you, Iris.
Okay, so can you hear me in the back? Yeah, because I tend not to be loud enough. So first of all, thank you very much for this opportunity to speak here, and also for the kind introduction. It's certainly a challenge, and I have never spoken where the biologist is the exotic person. Okay, so let's look at the complexity of the nervous system. So if you're a neurobiologist, you're absolutely utterly fascinated by neurons. They are amazing in their diversity and have elaborate shapes. And clearly, their morphology is telling us something about their role in connectivity in neural circuits. And neural circuits, as you know, in our brains can be incredibly daunting and complex. And that is illustrated here in this connectome of Vanessa and Fallman. And this one is just showing the connections among cortical areas associated with vision and vertebrates. So it's just areas. We're not talking about neurons yet. So in order to really understand and tackle the challenge of understanding the brain, we need to kind of find a balance between single-cell resolution and the complexity of neural circuits. And there are many ways how people tackle neural circuits, and one of it is actually, which we had chosen, and is to presumably turn to simpler model organisms, in particular invertebrate model organisms. And because complexity does not evolve all at once, but it's built up during development step by step, so therefore it is also a useful way to combine these two. So the complexity of the nervous system and of invertebrate model organisms, in particular the insect visual system, was already recognized more than 100 years ago by the grandfather of neurobiology, Ramon y Cajal. And in his biography he wrote, the complexity of the insect retina is something stupendous, disconcerting, and without precedence in other animals. And if you continue to read, he had a whole paragraph about how beautiful the insect visual system is. However, at the same time, he also recognized by studying the vertebrate retina how similar their organization is and how remarkable they both are in their organization in columns and in regular layers. And because of the similar functions these circuits have to perform, and also the similar structure, we strongly believe that we can actually learn from each other how the visual system works, how we can see. So our model organism is the Drosophila visual system, and is Drosophila and its visual system. And in particular, so the fly visual system consists of the compound eye. So 800 little eyes, subunits of matidia. And each ommatidium contains of 8 photoreceptor cells or short R cells. There are 6 outer ones are 1 to 6, and 2 inner ones are 7 or 8. Our 1 to 6 are the motion detectors, and our 7 and our 8 are the color detectors. Their cell bodies are located here in the retina, and they extend axons in the target area, which is subdivided in 3 areas: the lamina, the medulla, and the lobular complex. And I hope you can appreciate here the highly regular organization in cartridges, columns, and layers. So what are the building stones of the visual system in Drosophila? So as I said, so the photoreceptor cells are sitting here in the retina. And our 1 to 6 extend axons in the lamina, and our 8 and our 7 extend into the medulla. And the medulla is subdivided in 10 layers. Our 8 targets in the M3 layer, and our 7 in the M6 layer. So laminar neurons, I126 talk to laminar neurons within these cartridges modules, and each lamina in turn extends axonal arborization in a very precise pattern in these layers. And for example, this neuron L3 targets in the same layer as R8. And then from there, very simplified, the information goes to higher order neurons, the transmedullary neurons. And they connect the medulla with the lobula or the lobula and lobular plate. And then we need to have tons of intrinsic neurons, which connect higher layers and deeper layers or columns within one layer. So you think, what, this is pretty colorful and already complicated, but I can tell you this is just the beginning of it, because I want to give you like an insight about what numbers we are talking about. So this, in the eye, we estimate we have 6,400 cells, 8 subtypes. In the lamina, we estimate we have 4,000 cells, 5 subtypes. In the medulla itself, we have 40,000 cells and more than 60 subtypes. And in the lobula, we have 15,000. We don't know how many subtypes there are. So we're talking about 65,400 neurons per hemisphere. So to vertebrate brain, that sounds pretty good. But when we look at the complexity, so we can at least count on 73 neuron subtypes, and it's in the same ballpark as we would imagine, as we know is occurring in the vertebrate retina. So we are dealing here with a small yet complex enough circuit. So now if you combine that with the powerhouse of genetics in Drosophila. So you have a really very useful system to study how neural circuits assemble. Okay, so coming back to the columns and layers, because maybe as you guys, we also like to subdivide things in modules which we can handle. And so what is really quite striking is the organization in columns and layers. And why do we need them? They are developmental units because they need to bring the neurons together, which later have to make synaptic contacts together. And they're also functional units because the columns make it possible actually to represent pixels in visual space and to build a retinotopic map. And the layers are there because we need to extract out of each pixel different visual information, such as brightness, color, movement, and edge detected. So these units are really quite central to the functioning of the visual system. So now, how does a system like this develop? So the fact is that as everything, it has to go step by step. And this is like when we just look at a single neuron. So we have, first we need to specify a neuron subtype. Then this neuron has to extend an axon, and at the leading tip, it develops a very specialized structure called the growth cone. And the growth cone has the role to perceive repellent and attractive guidance cues like road signs in its environment, and it will help it to find. Eventually, it will reach its target area and recognize it, and then within the target area, the neuron has to find its postsynaptic partner, and sometimes they're just a part of this postsynaptic partner, and eventually it will engage to make synapses. So now if you build a circuit, what every neuron has to do, it has to go through this, and you have to coordinate this very precisely because it has to be built with a really amazing specificity. So that's modules or steps at a single cell level. But then when we look what needs to happen in the visual system, it is also quite clear that in bigger blocks, it also develops in steps. So first you have to make an eye where the photoreceptors are born and differentiate into clusters and so on, and they extend axons. And then at the same time in the target area, so you have to make the laminar neurons and the medulla neurons. The laminar neurons induced by signals from the eye, and the medulla is a diversity of the 70 or 60 subtypes, I think, is assembled by a temporal cascade of 5 transcription factors and, for example, notch signaling. And then the next step is the photoreceptors have just to choose, do I go to the lamina or do I go further into the medulla? And from our earlier work, we know that this involves interaction with another cell type in the nervous system, the glia cells. So now we just set up the basic cells and the basic projection patterns. And then during people development, everything gets reorganized. And now what the neurons are doing, they have to select their postsynaptic partners. And in particular, we have to form these laminar cartridges units. And in the medulla, one has to form columns and target and form these layers. So because we made progress, but we're still very much at the beginning. So what we need to do for choosing or studying a circuit, we just zoom into one neuron. And I just want to focus here on this R8 neuron and just ask, how does it get to this M3 layer? And again, so we need to look closer into development. And here you see some life-staining images during pupil development. In green, you see the photoreceptors. And here you would have the lamina, which forms the cartridges. But focusing on the medulla, you can see that first R8 axons and R7 are terminating in temporary layers, and R8 is right at the border of the nurple. Then you need to have a rotation of the medulla, and still R8 and R7 are in temporary layers. And finally, at 55 hours of pupil development, R8 proceeds to its final layer, M3, and R7 to the R6 layer. And in red, it's a marker flamingo, which allows us to visualize some processes of target neurons. And what I hope you can see is, at the beginning, we just have lots of columns on one layer. Then we have two layers. Then we finally, at 55 hours, we have the third layer, the target layer of R8s emerging. And what it tells us pretty much, we need to have this two-step targeting process because the photoreceptors have to wait until the target area is developing. Okay, so now if you focus and just ask, how does R8 get from here to here? So for today, just to give you an insight a little bit, we know that from other labs and our own lab that there are a couple of molecules which are involved in this process, like flamingo, a non-classic criterion, capricious, a leucine-rich cell adhesion molecule, golden gall, another transmembrane molecule. And in our lab, we identified an anterograde kinase signaling system formed by Jelly Belly and ALK. And to understand this further, so we did a candidate approach, and then we found a very classic guidance system formed by natrin and frazzled, which plays a key role. And natrin is a guidance cue, an attractive guidance cue, which is perceived by this guidance receptor frazzled, and in vertebrates it's deleted colorectal cancer and ANG40 in C. elegans. So I will not tell you the whole story. I will just give you a few pieces of information and then show you the model. So we started by looking at the expression of the receptor. And when you look in here in red, at early pupil development, you can see that the receptor is really closely expressed where R8 are terminating in the temporary layer. And then the receptor comes on more and more, and we start to see layers. In 55 hours, we see really a strong enrichment of the receptor in this M3 layer. And then we wanted to know, is it also expressed by the photoreceptors? And what you can see here, we looked in the pupil retina, labeled all photoreceptors here with this antibody in red, the R8 just in green, and in blue frazzled. It's a little bit faint, but I hope you can still see that at this stage of development, frazzled is only expressed on R8. So we have the receptor on R8 and lots of target neurons. And when we knock down frazzled through a genetic trick in the target area, we can see it's also enriched in the growth cones. So does it have a function? And one of the things we typically do is we try to remove specifically our genes of interest using a recombination system. And this one is called Eilers flip system. And the bottom line, what it allows us to do is to remove gene function from the eye, while the rest of the animal is heterozygous. And when we look at the phenotype, so we labeled R8 axons with RH6 like 0, just a marker, and we can see that all of them in controls terminate precisely in the M3 layer. However, if we remove the guidance receptor frazzled, we can see that the axons stay at the border of the nurple, and or they go halfway. So and the penetrance is very strong because we get almost 89%. So it must be a major player. Then from that, I make a huge jump and say, what about the ligand? And we want to know which neurons can make natrins. And for this, we look at enhancer trap 4 lines, and which activate membrane-bound GFP. And we can see that one neuron stands out, and that is this neuron I mentioned earlier, which is laminin L3, which has axonal arborization right where R8 is terminating. And then there are tons of other neurons. But it's a secreted ligand, and we need to ask, where is the protein actually? And when we look at the protein, so we use a knock-in of the cDNA in the endogenous locus using tagged with MYC. So we can use the MYC antibody to visualize where natrin B is. And when you look at early pupil development, it's very diffuse. However, already at 42 hours, you start to see one layer emerging, and at 55 hours, it is really screamingly strong. So now if you are a neurobiologist and you see your guidance cue, your road sign, like just exactly where you want to target your neuron, so this it cannot be more perfect. So here's the model to summarize a lot of data. So what we think is happening, so we have frazzled, which is generated and expressed by medulla neurons. And laminar neurons L3 release locally natrins and frazzled in the target areas, preventing natrin to diffuse. And at the same time, early pupil development, other guidance receptors, which we don't know, are holding a rate in place and say, don't go, we are not ready yet. And then at 50 to 55 hours, the block is released, and then the frazzled can be, expressing a rates can be guided specifically to this one layer. So for us, this is, it's really quite exciting because we could explain one strategy we uncovered one strategy which allows us to control one step of right axon targeting, the targeting to its final layer. And it's a basic fundamental principle because you use a chemo-attracting guidance cue which is localized and targets a neuron to a specific layer, and it provides positional information. So for people who are studying layer-specific targeting, that's a pretty, it's an important concept. And I would like to tell you that it sounds pretty daunting because now we just found one molecule class explaining one step in making this neural circuit. So frazzled clearly has more function. So it guides array axons to the target layer. It's involved for localizing natrins. It's very likely, we're studying this for the moment, also involved in guiding the partner neurons into the same layer. And one thing what we need to do to study and tackle this question is we need to know which neurons generate neurons or which require frazzled. And in circuit analysis, all the time what we need to do, we need to map the neurons. That's just in a circuit. That's the beginning. And for this, in flies, we have like libraries available, GAL4 drivers, stolen from yeast, introduced in flies, which allows us to express membrane-bound GFP reporters and label genetically cells of interest. But the problem is that even if we have lots of GAL4 drivers, they are not active most of the time in a single neuron type, but in many subtypes. And so what can we do about this? So we can chop up even more the short enhancer fragments. And so in Engineering farm, libraries like this are built. And we can use other genetic tools and intersectional techniques just to narrow down the expression pattern. So, but even if we would be successful to do that, we still have the problem that if we label a single neuron, in each nervous system there will be more than one. And what happens is that we have tons of green neurons and they all overlap and we still don't know what the single neuron is doing and what it is. So what we need to do is, and how we can help us, is to label things in different colors. And clearly, so many of you probably, or maybe some of you have heard about the ingenious rainbow method developed by Jean Livet, Georges Saint, Jeff Lichtman, and colleagues in 2007, which is the pioneering multicolor cell labeling approach in mice for the nervous system. And what we did in our lab is we wanted to build the fly equivalent, which we called FlyBo, and this was done by Daphne Hatchekunumu and our collaborators. And I feel almost embarrassed to say this, but this is like our little bit of synthetic biology at a minuscule scale. So, but how does it work? So both Rainbow and FlyBo rely on recombinases, which recognize specific sites and mediate target-specific, site-specific recombination. If you take the FRT sites or lock sites, and they're in the same orientation, and you activate the flip or recombinase, you would get an excision of the DNA fragment in between. If you put the the FRT recognition sites or lock sites in opposing orientation, then the flip or CRE will mediate an inversion. So that's the key idea for Rainbow and FlyBo. So we wanted to settle on flip recombinases because CRE is just not very tameable in flies. And we wanted to use a different flip recombinase system, which has a different specificities than the canonical one. And that is actually satisfied by the AMFLIP5 FRT71 system, which have very little cross-reactivity with the canonical flip FRT system. And what allows us is to actually plug in our cell labeling tool then with available flip FRT approaches. So here are the 3 FLIBO variants and their shared features. So we have FLIBO1, FLIBO11, FLIBO2. And I will go in a second how they work. And I will just highlight what we did here. And that is, so FLIBO transgene are based on the Brainbow2 strategy, which means they rely on excisions and inversions. So we use a 4 fluorescent protein, and all of them are membrane tethered. And one of them, serine, had to be epitope tagged. All constructs are under the control of the GAL4 US system that we can really genetically target any cell we want. And it uses, as I said before, a combination of the flip FRT and the AMFLIP5 MRFT71 system. And it's not shown here, but on top of it, we also make use of the ADP Phi31 integrase system to really put our transgene in location in the genome of flies we want, so that we can control for expression levels. So, and if you were to look, so pretty much we have like DNA pieces from phages to bacteria to viruses to like everything, to mice, everything is in there. And at the end, we end up with a functional transgene. So how does FLIBO10 work? So we have the core is like one cassette, which is consisting of a membrane-bound CD8 M cherry and a membrane-bound CD8 serine V5 tagged. And it's flanked by opposing FRT sites. So now if we express flip recombinase, which we induce by heat shock, what we get is actually, so the default is what is closest to the UAS site will be expressed, so we get lots of red cells. If we activate flip recombinase, we get an inversion and we will get blue cells. And that looks how it looks in real life. Whoops. So we have here in the third cellular stage red eyes and blue patches, and we have also blue neurons in the optic lobe. So the next stage is the FLIBO11, and in this case, what we have 2 cassettes. We have membrane-bound GFP, membrane-bound citrine, and cherry and serine in the second cassette. In this case, the default color will be green, and now we can, through expression of flip recombinase, we can trigger the following events. So we can have through an inversion of the first cassette, we can get imcitrine. We can have, through an excision of the first cassette, we can get a cherry. We can inverse the whole thing, or we can get rid of this one and have an inversion, we get blue cells. And then, so at the end, we end up with green, red, yellow, red, and blue cells stochastically. And that's how it looks like. So now, the same thing what I showed before. Now we have 4 colors. But here we're using a quite abundant neural driver. And what it shows us, that's pretty dense, and we will still have quite a lot of problems to reconstruct and recognize neurons. So that's why we developed a third system, which is the same as before, except that it has a flip-out cassette in front of it. And so now, flanked by a wild-type canonical FRT sites. Now what we get is no expression. But then when we activate both flip recombinases with heat shock, so we can excise this one, and now we are back to green cells and next to non-labeled cells, and then we can have the whole cascade, and we end up with non-labeled cells, and green, yellow, red, and blue. And that's how it looks like. So we achieved through this sparse labeling. And like this, so we really reach the level to reconstruct the neurons. So to come back to an example I set out with the natrine. So for us, it was really important to study which cells are expressing natrine. So we had a GAL4 driver, and we combined it with the FLIBO2 approach, and we could map now all the neurons. And obviously, we got lots of laminons L3, and then we got lots of other neurons like TM3 and TM20, and here we used another approach. And then so we got all these neurons through our mapping, and I'm totally aware that these numbers don't mean anything to you. And but the bottom line is that it helped us to actually discern that the only neuron which had actually axonal labyrization in this layer was the laminar L3, and everybody else had dendrites in this area. And then, so this helped us in principle to come to the proposition that the reason why we have this tight natrine layer and is because natrins can only be released by axon terminals and in particular by laminar L3 axons. So that's just to give an example how you can use this approach. Where do we go from Flybo? Because obviously it was a beginning and far from the end. And so we need definitely to improve the functionality, and there we benefit from the development of new fluorescent, brighter fluorescent proteins for the cyan and for red, and in particular mTurquoise2 or mTurquoise, mTurquoise2, and TAG-RFP. And this family for mTurquoise is shown here, and now we can actually really image the endogenous fluorescent signal and combine this with an antibody. So we are actually at the level where we can do 5 color labeling. So, which brings me to my summary. So I hope that I could give you an idea about the complexity of our nervous system, telling you about how the Drosophila visual system develops, and what approaches we can take to overcome the complexity of neural circuits, and that is that we subdivide development into steps. And our prediction is that each of them are regulated by specific genes and molecules. So now I set it up already a little bit earlier, but I gave you only one example to show you like one step one neuron needs to do and one guidance system. And it seems like in order even just to make layers, there isn't. Every neuron has kind of a cell-type specific program. And for that, we need to kind of ask, how many fundamental principles and cell-type specific programs do we actually need to uncover to build such a system? So which means, can we design it? As Lotte, I would say, big question mark. We still need to learn a lot. But on the up note, when I hear the talks today, it fills me a little bit with jealousy, but also with absolute delight, because I know. So for us, genetic tools such as Flybo, they are essential because our tools, genetic tools, will help us to analyze, define genetic cell population during development and in adults, and with single cell resolution in a circuit. And Flybo has been generated painstakingly by classical molecular biology. And when I see what synthetic biology can do, so I'm really very excited, because for a Drosophila developmental neurobiologist, synthetic biologic techniques will open new, previously unimaginable doors how we can design our genetic tools. And with that, I finish. No, sorry, off. I've never done this before. So, big credit, of course, goes to the people in my lab, in particular Daphne, who developed all the Flybo reagent, and really Julie, Katerina, Timofey, Nana, Shimosaki, who work on Natchin and Fuzzled, and of course our collaborators. Now I'm finished.
Hi, the neuronal circuit, the neuronal system that you presented at the beginning is probably one of the most complex biological systems known in nature. But every cell has the same genome. How many genes are involved in the development of this, of the Drosophila genome?
It's so for the target area, we don't know. However, for the eye, it is estimated that 4,200 of 13,800 genes are involved just to make the eye. So,
so it's phenomenal that you can do this by using multiple colors. If you could think of a toolbox that you would want to be able to do this, how many colors would you actually want to be able to label things with? Or how many independent markers would you want to be able to really do this at high density?
So that's a very good question, because obviously the rainbow mouse set are huge standard, because their tools can generate about 110 shades or hues in one go. We're a little bit more conservative here because we think that for the microscopy and the density of the processes, like even already 4 colors and some sparse labeling does a really good job because at the end, so we hit the block that we need to able to image and analyze it and to get the resolution. And then so the problem is that the neurons, so a tissue is thick. If you have multiple colors to get different shades, you will get changes of intensity. So to trace a complete neuron, so it is still a huge challenge. So I think maybe the double of the colors or something like this. But microscopy and computing need to kind of keep up with that.
One more question. No? Well, if there are no further questions, join me in thanking Iris for this talk. Good job. I think one of the spectacular things about this sort of work is it's beginning to give us a dynamic and spatial view of how complex systems organize themselves. And you might think this is only happening in the Drosophila brain, but of course it occurs even in bacterial systems. You look at them in soil or in Yellowstone Park in the mats. They're very organized, very complicated, and it's those contextual clues that make the complexity of engineering for things beyond the bioreactor that much more difficult. And so having tools allow us to trace biological determinants of development and behavior alongside the functioning of our circuits is going to be absolutely key. This is a wonderful example of how to do that. Our next speaker, Jason Chin, has been amazingly prolific in taking control of one of the most central processes in biology, and that is translation, and using it both to generate orthogonality between ribosomes, but also, very importantly, to use them to modify proteins to give them a whole new sets of functions they would not occur in nature, and then use them both to dissect biology and to create new enzymes and new technology. He's been really fantastic. So he is currently the program leader at the Medical Research Council Laboratory of Molecular Biology. He's also the head of the Center for Chemical and Synthetic Biology. He is joint appointed at the University of Cambridge Department of Chemistry, is a fellow and director of studies in the natural sciences at Trinity College. He's been through Oxford, a Fulbright at Yale, Damon Runyon at Scripps, and has won multiple awards, the Francis Crick Prize of the Royal Society, Chemistry's Corday Morgan Prize, the Embo Gold Medal, and so on. So a wonderful anchor for this session. Jason, welcome.
Okay, so thank you, Adam, for that kind introduction. It's a real pleasure to be here today to tell you a little bit about the work that my lab has worked on over a number of years that really focuses on a central paradigm in molecular biology in which the heritable information of DNA is copied to RNA, and then the triplet codons within the RNA are decoded to produce proteins composed of the natural 20 amino acids, the common natural 20 amino acids that form the proteins that carry out all the functions that we know about in biology, many of the functions. And so we've been interested, actually, since I was a postdoc with Peter Schultz, in understanding the extent to which we can reprogram this paradigm in molecular biology such that the heritable information of DNA is ultimately used to produce proteins containing new types of amino acids that we can chemically synthesize in the laboratory that go beyond that set of 20 that nature normally uses, and ultimately think about whether we can reprogram the processes that convert the information in the triplet codons in RNA to proteins to produce ultimately entirely unnatural types of polymer. And this is driven in part by the realization that the ribosome, the machine that does the translation of the RNA into proteins, the amino acid polymerase,
template-driven amino acid polymerase, is really an amazing paradigm for how to build up polymers of defined sequence and composition. So you have a nucleic acid sequence where a triplet codon defines the start, a triplet codon defines the end, and the sequence of non-overlapping triplets define the sequence of amino acids that are put together in a polypeptide. And we're interested in asking, can we define approaches to incorporate single amino acids, single new types of amino acids that are limited by what we can chemically synthesize and imagine in the laboratory, multiple identical amino acids, multiple distinct amino acids, and ultimately think about using this as a paradigm for synthesizing entirely new types of polymers, and these might be materials or second and third generation therapeutic type molecules. So over a number of years, we've worked on how we can, in bacteria, design systems that allow us, or create systems that allow us to go from making proteins to making proteins containing new types of amino acids, multiple types of amino acids, multiple different amino acids, and ultimately go towards creating completely unnatural polymers. And at the same time, we've developed approaches that allow us to take the ability to incorporate amino acids into proteins at defined sites from bacteria into eukaryotic cells, and most recently into whole animals. And I'll try and tell you a little bit about both of these directions today. So the first thing we need to look at is really, I've told you that what we want to do is to reprogram how information is read in the nucleic acids and used to make polypeptides. And this really comes down to the process of translation, which is shown schematically here. You have amino acids, and these amino acids are recognized by enzymes in the cell, amino A cell tRNA synthetases. And these amino A cell tRNA synthetases also recognize a cognate tRNA, and their role is to recognize a specific tRNA out of the set of 20 set of isoacceptor tRNAs in the cell, and a specific amino acid, and essentially covalently attach this amino acid to the tRNA, which sets the correspondence between the identity of this amino acid and the identity of the anticodon of the tRNA. And this is the process of amino acylation. It's then the process of decoding, where this amino acylated tRNA is taken to the ribosome, and this anticodon, via Watson-Crick base pairing, recognizes a corresponding codon in the messenger RNA, which is bound to the ribosome, shown here in yellow. And if this is a cognate interaction between the anticodon of the tRNA and the codon in the mRNA, then you get a peptide bond formed between this amino acid and the nascent polypeptide chain, which is already attached to the tRNA in the so-called P site of the ribosome. And then subsequent translocation resets the system and basically allows you to polymerize a sequence of amino acids which is encoded in the sequence of codons that are transited through the ribosome on the mRNA. And so if we want to reprogram translation to put new amino acids into proteins, there are really at least two things we need to do. First of all, we need to figure out how to put new types of amino acids onto tRNAs, and this is going to require finding amino A cell tRNA synthetases and tRNAs with very particular properties. And we also then have to find codons that will allow us to incorporate those amino acids uniquely into proteins, since we need to find additional codons with which we can encode the incorporation of these new amino acids beyond the codons that are used to encode the set of amino acids that are used in natural biology. And so altering the translational machinery itself is incredibly difficult because you might know that, of course, the translational machinery is very well conserved between cells and between different types of cells and organisms. But moreover, the ribosome and synthetases and tRNAs in cells are essential for making the proteome, the complement of proteins that functions and keeps the cell alive. And so any mutations that you make to the translational machinery are often problematic, lethal, or dominant negative. And so this is a central question in how we think about engineering very central and fundamental things in the cell. If they're essential, you're in a sort of catch-22 situation where you can't engineer them, but these are exactly the things you'd like to be able to engineer because they potentially would allow you to alter some of the most central functions of the cell. So we did initially a thought experiment. We said, well, if this is what natural translation looks like schematically, you have natural ribosomes, natural messages, natural tRNAs that use natural amino acids. We said, what would happen if, in fact, within the cell, we were able to build a new ribosome that read a new message, okay? And that ribosome had very specific properties that operated in parallel with, but independent of the natural ribosome, okay? The reason that this system is unevolvable is because we can't make mutations because they interfere they're likely to interfere with the synthesis of the entire proteome. But here we've got a ribosome directed specifically to a new message, and so any mutations we make in this version of the ribosome really only interfere with the translation of that one message. Okay? So we've uncoupled, we basically make a new version of the ribosome which is uncoupled from the original ribosome, and our hypothesis is that that ribosome would then be evolvable, that is, we could alter it to do new things that we can't do within the context of the natural essential ribosome. We also realized that the genetic code itself is really just a set of correspondences that you can write down in a table. It's a set of correspondences between the identity of particular codons and the identity of particular amino acids. And that's really set by the translational machinery. tRNAs come into the ribosome loaded with the amino acids and decoded in response to the corresponding codon in the messenger RNA. But actually we realized if we could deliver tRNAs selectively to this ribosome, then we were free to rewrite the rules of the genetic code and deliver amino acids in response to any codons, and in fact alter the rules by which the ribosome itself reads new codons. So from this thought experiment, we got the idea that if we were able to make a new parallel independent ribosome in the cell, that ribosome would be evolvable. And one of the things we might be able to evolve it to do would be to read the genetic code in new and different ways. And so we thought this would be an interesting thing to work on. And so over a number of years, we've addressed some of the challenges in building this parallel independent translation system for incorporating unnatural amino acids into proteins, unnatural monomers into proteins. And I'm going to walk you through some of the challenges or some of the strategies that we've taken to do this. And this really involves building particular modules that have particular functions, and then putting those modules together to build a new translation system. So the first question is, how do you amino acylate a tRNA with new unnatural monomers? Then the second question I'm going to address is, how do you specifically direct a version of the ribosome to a new message and not to endogenous messages, and create a message that's not a substrate for endogenous ribosomes? And then in terms of being able to build this parallel genetic code, how do we selectively deliver tRNAs loaded with new amino acids to this new ribosome mRNA complex? I'm going to take you through each of those challenges and how we've gone about, in outline, addressing them. So the first challenge is really how you get a new synthetase tRNA pair in the cell that recognizes a new amino acid and puts that new amino acid into proteins in response to a new codon. And the challenge is that within the cell, there's a set of 20 synthetase enzymes, 20 amino acids, and a set of 20 sets of isoacceptor tRNAs. And with respect to those, what we need is a synthetase that doesn't load any endogenous tRNAs with its amino acid substrate, a tRNA that's not a substrate for an endogenous synthetase, and then we need the synthetase to recognize an entirely new substrate that biology may not have seen before. And it turns out you can solve this problem in two steps. You can first take a synthetase and tRNA from an evolutionarily divergent organism. This is taking advantage of natural evolutionary divergence, and find ones where they don't cross-acylate the endogenous tRNAs in a host and where the tRNA is not a substrate for the endogenous synthetases. You're then left with the challenge of how you alter the active site of the enzyme, since this enzyme will typically, all cells effectively use the same set of 20 amino acids, you're left with the challenge of how you'll reprogram this active site to recognize this new substrate. And it turns out that you can do this by using a series of directed evolution approaches where you make large libraries of variants in the active site of the enzyme and find those variants that recognize, on the order of 10 to the 9 variants, and find those variants that recognize this new substrate, but no longer recognize any of the natural amino acids. The second challenge then was how we readdress the ribosome to a new message. So the ribosome in bacteria, or in E. coli at least, recognizes the messenger RNA via a so-called Shine-Delgado interaction between ribosomal RNA and the small subunit of the ribosome and the leader sequence in the messenger RNA. And so we made, again, a second copy of the ribosomal of the messenger RNA, a second copy of the key bit of the ribosomal RNA. And within each of those, we made targeted libraries, mutagenic libraries, where we asked for messenger RNA sequences that were no longer substrates for the endogenous ribosome, and ribosomes that no longer worked on endogenous messages, but were able to translate the new selected messages that are not substrates for endogenous ribosomes. So this got us to the point where we had two sets of ribosomes that we could express in the cell, one set of ribosomes that keeps the cell alive and has always been there, and a new ribosome which is now directed to a new message, and in principle we believed that this ribosome should be, we ought to be able to alter or evolve this ribosome in the cell. Moreover, we realized that this ribosome is now a non-essential ribosome, and so we ought to be able to use it as a new sort of regulatory element in the cell. And since you can't use ribosomes naturally as regulatory elements, this opened up new opportunities. And one of those opportunities that I'd like to briefly diverge to tell you about is the idea of making entirely orthogonal genes in the cell where the transcription and translation of those genes is not carried out by the host protein transcription and translation system, but is carried out by an orthogonal system, an orthogonal ribosome, and an orthogonal, in this case, RNA polymerase that you add to the cell. So this raises new opportunities in terms of the way that you can regulate gene expression. So now, for example, we can make the expression of a gene and the production of a protein dependent on both, as an AND function, dependent on the transcription and translation of the gene. And since the ribosome that translates that gene is non-essential, we can turn that on, so this is a true AND function. We can also express the ribosomal RNA from the polymerase, and this is, if you like, a feed-forward system where the T7 polymerase drives expression of a ribosomal component and drives expression of the transcript that then that ribosome will translate. And it turns, I won't go into the details, but it turns out that this allows us to generate ways of regulating the timing of gene expression that are different than those that natural biology could use. Having developed a ribosome that's directed to a new message, I've shown you that we can use that as a regulatory entity, but we really wanted to know whether we could actually evolve this to do things that you can't do with a natural ribosome at the level of molecular function. And one of the challenges in incorporating unnatural amino acids was the fact that these unnatural amino acids were being directed into proteins in response to what's called the amber stop codon, which is normally actually recognized by a release factor and leads to termination of protein synthesis. So we were competing, this amino acid incorporation event was competing with the release factor. And the termination leads to a truncated polypeptide, whereas the decoding of the tRNA leads to the incorporation of the unnatural amino acid. And so we said, can we actually evolve our version of the ribosome such that it no longer recognizes release factor and efficiently decodes the tRNA by virtue of no longer recognizing release factor? And this would show that both that we could evolve the ribosome in the cell and provide a useful approach for more efficiently incorporating multiple versions of the amino acid into a single polypeptide. And indeed, we were able to make structurally directed libraries in this new version of the ribosome and evolve a version of the ribosome where we've now got our ribosome that's able to incorporate unnatural amino acids much more efficiently than protein translation on the natural ribosome. So in this experiment, we were able to show that you can differentiate the way that two translation systems in the cell decode the same genetic information. We went on to ask, well, if we can evolve the ribosome to read new genetic information, can we get it to read codons that are not even triplets? Can we make a ribosome that can read a quadruplet rather than or in addition to triplets? So again, the natural ribosome and our ribosome are both inefficient at decoding extended anticodon tRNAs to read quadruplet codons. And again, we made structured guided libraries in the region of the ribosome that decodes the tRNA, and we're able, via a directed evolution approach, to select a ribosome in the cell that's able to efficiently decode a quadruplet codon and incorporate an unnatural amino acid in response to that codon. And then by taking synthetases and tRNAs that recognize distinct amino acids that don't cross-react with each other, we're able to now begin to direct multiple different amino acids into a single polypeptide, taking advantage of the fact that there are a large number of quadruplet codons that we can read on this messenger RNA and use to direct uniquely the incorporation of unnatural amino acids. And in principle, there are up to 256 such codons that we could reprogram using this approach. Our initial approach has been to actually just reprogram 2 of these, and this is primarily limited by the number of mutually orthogonal synthetases and tRNAs that we have to direct the incorporation of distinct amino acids. But this allowed us to put together the modules that have been developed for putting the synthetase tRNA amino acid modules with the ribosomes and the evolved ribosomes, such that we could differentiate the genetic code and direct a translation pathway selectively onto this orthogonal ribosome and orthogonal message and efficiently incorporate multiple distinct amino acids. And one of the things this has allowed us to do is to ask, can we now build systems where we program into proteins amino acids that can interact in ways, in new ways? So this might be 2 amino acids that are genetically directed that interact and transmit a fluorescent signal from one amino acid to another, or you might direct the formation of a covalent bond, which was the first thing that we decided to do, to actually think about how you can covalently staple proteins by methods other than, for example, disulfide linkage, which are redox, to make new linkages which are redox stable. And we're interested here in developing approaches for making, for example, stabilized versions of protein therapeutics. So up to this point, I've told you about, primarily in bacteria, about how we can take the natural genetic code and how we can fundamentally reprogram some of the key components of the translation system in bacteria. And now what I want to tell you about is how we can take this paradigm of creating orthogonal synthetases and tRNAs for putting amino acids into proteins, again in response, going back to the AMBER codon, and advances in using this, in developing systems that allow us to take this approach from bacteria and into eukaryotic cells and most recently animals. And most of this work has really taken advantage of a synthetase in tRNA that comes from certain methanogens. And these methanogens use this unusual amino acid, pyrrolysine, and they put that amino acid onto the tRNA using a synthetase and incorporate that amino acid in response to the AMBER stop codon. And this synthetase in tRNA is orthogonal in E. coli. That is, it doesn't cross-react with the endogenous synthetases in tRNAs in E. coli. And we've done a variety of experiments showing that we can incorporate a range of unnatural amino acids into proteins in E. coli, make recombinant proteins, do structural biology, biophysics, entomology, primarily directed at understanding the role of post-translational modifications in regulating protein structure and function. But I'm not going to tell you about that today, because what I want to focus on is the enabling ability of this pyrrolysyl synthetase in tRNA to allow us to move seamlessly from bacteria to eukaryotic cells. And this is a major advance in the field, because previously we had synthetases in tRNAs that allowed us to put new amino acids into proteins in bacteria, but we couldn't use these synthetases in tRNA in eukaryotic cells because they cross-reacted with the endogenous synthetases in tRNAs in eukaryotic hosts. Similarly, we had synthetases in tRNAs that we could use in eukaryotic hosts that were derived from bacteria, but those we couldn't use in bacteria because they cross-reacted with synthetases in tRNAs in bacteria. The great thing about this system is that it's orthogonal both in bacteria and in eukaryotic hosts. So you can do all the engineering and testing of the active site of the synthetase in bacteria, where making large gene libraries and doing those sorts of experiments is more straightforward. And then you can simply transfer the information that you get from the bacterial system into eukaryotic cells and most recently eukaryotic animals. So this has really accelerated this ability to make systems that you can develop in a simple system and then use transferably, has really accelerated progress in this field. What types of things are we really interested in being able to do? We're interested in being able to generate new ways of regulating protein function. And one of these ways that I thought I would highlight for you is shown here. So here's a natural amino acid, lysine. And this bit of the compound that I'm showing you here is a photocaging group. So we can evolve a synthetase, a pyrrolysyl synthetase variant to recognize this amino acid in bacteria. We can transfer that to eukaryotic cells, mammalian cells, and we can encode this type of amino acid into a defined site in a polypeptide in a mammalian cell. The trick is now that when we shine light on this, this whole bit comes off and we reveal the lysine side chain. So if you have a protein where the function of the protein depends crucially on a lysine side chain, you can mask that lysine side chain and rapidly activate the function of that protein with light. So to give you an idea, you can do this decaging in milliseconds. So this is an approach for photocontrolling protein function with millisecond time resolution and very high spatial resolution. The first type of experiment we did here was really to fuse this to a nuclear localization sequence that contains lysines, and this blocks the function of the nuclear localization sequence. And now the hypothesis is when we shine light on the cells, we'll be able to relocalize the protein to the nucleus. So this is as a fusion to the green fluorescent protein, and some of this protein goes into the nucleus anyway. But hopefully you'll be able to see when we run the movie on one of these panels, at time 0, we do the flash, which is a millisecond pulse of light, and you'll see the protein rapidly move into the nucleus because we revealed the nuclear localization sequence, and we can follow this very reproducibly in cells, so this is a way of controlling very rapidly transport processes. We also wanted to be able to think about how we could use this approach to dissect and control and manipulate signaling networks. So here's a textbook sort of view of MAP kinase signaling. Typically, you might activate the receptor, a whole series of processes, phosphorylation events might happen, and then MEK will phosphorylate ERK, and you might read out, for example, ERK moving into the nucleus. But really what we'd like to be able to do is to be able to turn on these pathways at any points, so at any point in the pathway to be able to create subnetworks so that we can dissect out the contributions of elementary steps in signaling, the role of feedback regulation, etc., and be able to, for example, activate a single kinase in this pathway. So, for example, if we could rapidly activate MEK, independent of receptor stimulation, then we could study the network that goes from MEK to ERK to the transcription factors in the nucleus and really try and understand what the kinetics of that process are and how that relates to feedback mechanisms that operate in the cell. The way we decided to do this is a near universally conserved lysine in the active site of the kinase. We replace this with a photocaging group and introduce mutations that make the kinase otherwise constitutively active. So now when we shine light on the cell, we can rapidly photoactivate the kinase. So what can we learn from these types of experiments? Here's an experiment where we stimulate the cell with growth factor, and what we're watching is fluorescently tagged ERK moving into the nucleus. So you see there's a lag phase, then ERK accumulates in the nucleus, and then you have an adaptive dissipation of ERK out of the nucleus. How does that compare to the situation when you just rapidly activate MEK? This is what you see. You lose the lag phase, so that tells you presumably that the lag phase comes from steps upstream of MEK in the pathway. Then you have a very rapid accumulation of ERK in the nucleus, and you lose this adaptive response. So this tells you something about where the steps in the pathway where players must be acting to cause this adaptation, i.e. it must be acting at or upstream of MEK. So you can imagine walking through pathways and getting not only synthetically controlling pathways, but getting information about how those pathways are naturally controlled. This is what the primary data looks like. Again, I'd like to emphasize these are millisecond pulses of light, and we can follow the processes. We can follow these processes, in this case, over many minutes. So really what we're doing is making perturbations that are on a much faster timescale than the biological process we're studying. And this is a way that, if you can think about this as being a way that you can, by embedding new chemistry into proteins, you can generate types of regulation that it's hard to generate using biological entities alone. Because we're interested in taking these approaches and applying them to the study of things that are very hard to address in cells, mainly neurobiology, development, and disease progression, we're really interested in moving these approaches into animals so that we can develop approaches with this sort of molecular, spatial, and temporal precision, but within the context of a very complicated system. And we've recently done the first work on this with Mario De Bono at the LMB in expanding the genetic code of C. elegans. And again, this really comes down to the fact that we're able to develop a lot of the basics in E. coli and then transplant these systems into, in this case, the worm C. elegans. And so the final data slide I'll show you is really the first example of an animal, a free-moving multicellular animal that has new chemistry embedded in its genetic code and the ability to encode more than 20 amino acids in its proteome. And you can see that by virtue of the fact that these animals make, in this case, a variety of types of data, but in this case, we're showing that they make this M-cherry protein as a function of incorporating the unnatural amino acid into the protein. We've also recently done experiments in flies, which have the real advantage, as Iris pointed out, that you can genetically target the expression of these amino acids to particular cells within the whole animal very easily using GAL4 and other systems. So what I've tried to tell you about is work in my lab that's gone from incorporating single unnatural amino acids into proteins to incorporating multiple different types of unnatural amino acids into proteins by thinking about how we can engineer the translational machinery as a whole and work that's been driven by trying to develop approaches to study, develop new approaches and new tools, including imaging approaches that I didn't talk about, but also photo control approaches that I did talk about, to move these approaches into mammalian cells and into animals. And so I'd like to thank really everyone in my lab and the funding agencies that have funded this work, and you for your attention.
Questions? Here.
Thanks, Jason. That was a great talk. Obviously, the ribosome is a multi-subunit complex, and presumably you're modifying multiple different subunits. So I just wondered, in terms of the ribosome assembling and being functional in the cell, do you end up with chimeric ribosomes? Presumably you can't control...
No, so the primary functions of the ribosome, well, the first approximation, the primary functions of the ribosome are in the ribosomal RNA. And so everything that I showed you so far is involved with engineering the ribosomal RNA and taking advantage of all the other components, the protein components, for example, from the endogenous pool. So there's no problem scrambling.
But can you control all those modified ribosomal RNAs always ending up in the same ribosome complex?
In this experiment, there's only one ribosomal RNA that's been modified.
Oh, okay,
yeah.
Right.
There's only 3 or 2 in total, so yeah, depending on where you are.
Hello. Yeah, there we go. Sorry, I'm up here. When you're using quadruple nucleotides instead of triplet nucleotides to encode your codons, how, when that quadruple nucleotide, when it moves into your ribosome, do you stop a triplet nucleotide from recognizing it? And then you're getting a frame shift, if that makes sense.
So you have an intrinsic competition between a quadruplet. I think what you're asking is you have an intrinsic competition between a quadruplet decoding event and a triplet decoding event, where the triplet, for example, might be the first 3 bases of the quadruplet.
Yeah, yeah.
Is that what you're asking?
Yeah, that's right.
So we only have quadruplet decoding tRNAs for the quadruplets that we want to decode. And because we're writing orthogonal messages, we can leave the corresponding triplets out of the message. And we believe, though we haven't rigorously shown this, that the quadruplet decoding efficiency is more efficient when we have a quadruplet there. Hey.
Talk about perhaps the challenges and progress towards engineering the 50S subunit and the peptidyl transferase center to be orthogonal in cells.
Right. So what I've shown you today is really based on engineering the 30S, the small subunit of the ribosome. And so the question is really about the large subunit of the ribosome. And whereas the mRNA recognition and the tRNA decoding, to a large extent, occurs in the small subunit of the ribosome, the peptidyl transferase center of the ribosome is in the large subunit of the ribosome. The peptidyl transferase activity is in the large subunit of the ribosome. And that controls the types of chemistry that you can access in peptide or other types of chemistry bond formation. And so one of the clear challenges here is that what you'd like to be able to do is obviously to be able to recruit a version of the large subunit, specifically to the orthogonal version of the small subunit, so that you have a complex that works in unison. And the hypothesis is that that would allow you to evolve the large subunit to do a variety of things, recognize the FTU differently, do different types of bond-forming reactions, et cetera, and you could do selections on all those types of things. The experimental challenge in that is that the ribosome ribosome interface between 2 subunits of the ribosome is primarily RNA-based. And it's a 6,000 square angstrom RNA-RNA interface, which is much larger and quite different from anything else that people have really thought about or tried to engineer when you think about engineering small protein-protein, well, I shouldn't say small, but protein-protein interfaces. And other types of RNA-RNA interactions that might be dominated, for example, by base pairing. This is primarily dominated by tertiary RNA-RNA interactions. Moreover, while we have crystal structures of the ribosome, in part from my colleague Venki Ramakrishnan and others, these are static structures of one functional state of the ribosome. And we have lower resolution structures that tell us a little bit about the dynamics, but what we don't really understand in molecular detail is how these things rearrange. And if we want to engineer them, we've got to think about not just engineering them to work in one functional state, but being able to engineer them so that they work across the series of states that they ratchet through. And that's a combinatorially large and complicated problem.
Okay, I think that's all we have time for.
Okay,
Thank
great.
you, Jason. It was wonderful.
More questions.
So we're going to move into the next phase of this, which is a panel discussion. And I'm going to take some privilege to set some context. I want to reflect a moment on what the speaker said. The challenge of the session was to understand how to make more efficient the design, build, test cycle such that we could build more and more complex function. And we covered a wide range of topics that touch on this, but with the exception perhaps of Chris's talk, and Richard's talk, didn't directly get to this yet. What was interesting, though, was the following. We had one biologist struggling with how to factor out a very complicated physical system in a microbe, which required understanding how not only do the proteins regulate themselves, but how they do so in space and in opposition with the compartment in which they reside. We had another one who used very sophisticated DNA editing operations to try to dissect how complicated populations of cells developed and became connected in the right ways. And we had Jason, who talked about how to co-opt the biological mechanism and turn it into something that increased the functionality of the system and to create entirely new organisms, in a sense, with new functionalities that he could use both for pharmaceutical purposes but also for dissecting complicated pathways. On the other hand, we had people trying to build new ways of optimizing the systems so that they would meet our specifications. What we didn't hear as much, I think, was what we were specifying for. We know, with the exception of nitrogen fixation, that we have certain goals, but what are we trying to build that requires complex design? So let me talk a little bit about that. Can we have my slides up for a moment? No synthetic biology conference is complete without a definition. There are as many of them as there are of us, but they all sort of revolve around this. And I want to talk a little bit about why I think this is a particularly interesting definition. One is, it's a field dedicated to making the engineering of new complex biological function vastly more whatever. But the point is, it's not about the applications. It's about the process of making engineering more efficient. Applications are important, though, because engineering is only important when applications are present. Complex is the other one. Now, that's a judgment. I think complexity is interesting. The reason why I say that is because since Asilomar and before, we have made less complex things easier to engineer. It was on the roadmap in genetic engineering. Make it easier to express proteins, make it easier to express pathways, make it easier to express new functions. What's new is complexity. These other words here are about transparency, meaning understandable, which is why evolution is a module of the field, but not the field itself. We need to make what we do understandable to each other. Efficient, obviously, reliable, predictable, obviously, and safe. These last three are about trust and credibility, both amongst ourselves, that what I designed will work for you, and with us and the public, that what I designed will operate as I say it will operate and not any other way. So if you think about systematic engineering, it's really about these blue words here and making that work. So what are the issues? And I always have to alliterate. I have my 4 C's here: construction, courage, context, and credibility. Construction. That says, do we have the parts to construct with? Can we put them together? Can we insert them effectively and stably, etc.? I'm going to argue, perhaps controversially, that I'm not worried about this anymore. I'm not. Things are scaling now here. we got parts. This is just DNA sequence. We're able to sequence new things very rapidly. There's 549,000 eukaryota by phylum with sequence in GenBank. There's 226,000 bacteria. This is not fully completed genomes. This is just pieces of sequence. But in my lab, I do about 96 genomes a week. It's not a big deal, right? But, it's just a lot of different things. And we have many orthogonal families of regulators for key organisms, poorly characterized. Approximately 300 enzymatic activities of interest that we need for industrial things, poorly characterized. And a pitiful number of sensors and other actuators, such as nitrogen fixation, motility, spatial control. Pitiful, actually. Also, poorly characterized. So what's the roadblock here? It's not the parts, it's not the assembly, it's characterization. Can we put them together? Can we insert them a lot? Everyone knows who this is. This is Cynthia, obviously. This is Craig Venter's, fully booted genome. My point is that at the point where we can do this, yes, it's hard and it's expensive and all this, but I'm not worried about this anymore. We can do it. It's not an issue for me. So what are the issues? Courage. This, for me, is an issue. Drew Endy said last night, the field's becoming boring. I'm sure he didn't mean it exactly that way. What he meant by that is the things we're approaching are still at this modest level, and we're getting better and better and better and more mature at it. But what's the next phase? How can we go beyond the bioreactor and completely change things? So are we designing what we really want to build ultimately, what the world will need ultimately? I want to point out a study. This is the 2012 World Economic Forum of the Global Risk Report. This is an interesting graph. I like two-dimensional graphs, as you'll find out. Not 16-dimensional. We have here the likelihood of a bad event and the impact of that bad event according to this august council. I've circled the things where I think synthetic biology is directly interesting. There's a bunch of other things where it's indirectly interesting. Water supply crises, food shortage crises, extreme volatility in energy and agricultural prices, rising greenhouse gases, and so on. These are all things with biology in the loop, for which biology is not only a key element of what's being affected, but what can affect an outcome. There's a lot of them there. We've seen a lot in the last week on antibiotic resistance from J.J. Collins, for example. We've seen a lot about how to make things better for chronic disease recently, especially from people like Carl June and the adaptive immunity. We've seen bioenergy becoming a big deal. But how are we going to make all of this work when a lot of these things have to do with agriculture? And people said avoid food last night. Someone said we should avoid food. No, we shouldn't avoid food. But I want to think about that. What does it mean? So is biology the right thing to do in the loop here? By the way, this council called out the top 10 technologies that were going to save us. You'll be happy to know we're number 2, along with metabolic engineering, which they separated out, by the way, as separate from synthetic biology. Keep it in mind. The Green Revolution 2.0, systems biology, computational modeling. We've seen a lot of systems biology today. And personalized medicine, which essentially is systems biology with an application. These are things we have to deal with. Informatics, something we've talked about a lot. Now, again, getting back to two-dimensional graphs, here's where I think we are. This is my two-dimensional graph for synthetic biology. It has 2 axes, discovery and design, and pipes and programs. So a discovery, a natural program is things like the yeast that make ethanol for us. Someone said last night, beer is our next big thing. I think it is, but a lot of the cells that we use that are really efficient at producing ethanol, we have no idea how they work. We can't tame them in the laboratory very well, and they're just found programs. They're very complicated reasons why they work very well in the big bioreactors. We also have discovered pipes, enzymes, just turn one thing into another, and those are an example, like the novozymes and the glucanases that are an example of these, although they've been optimized in the laboratory to some degree. We have maybe one really good example of a completely de novo designed enzyme. David Baker did that. And that's a pure design, but it's still kind of a pipe. It makes a retroadral reaction. Some small demo networks. And up here is where we have some problems. The cells that should be engineered soil microbes to enhance crop growth, to cell-based therapies, things like this. And the problem is that we have to get from here to here in a way that the public trusts us. And we trust ourselves to get it done. And we struggle with this. I think we saw a lot of complexity as people tried to understand what it would take to get here. Right now, most of us are sitting over here in modern metabolic engineering. It's where the industries lie. It's where the knowledge lies. It's where some of the value lies. I think we need to move beyond there, and we're sort of stuck with this application design barrier, trying to get across this thing with enough of us working together to get to these verified custom cells. So what's the issues of getting there? And that's all about context. The actual function isn't as hard as it seems. It's hard. What's really hard is dealing with, does it work in a changeable environment? Chris's 16-dimensional surface has a function of time and condition on it as it changes its shape. How does that work? It depends on the host. And we saw Jason trying to struggle with how to get things to work in eukaryotes and in prokaryotes and so on. It matters how things are connected together, because you put two things on the same piece of DNA, as Chris was showing, you can get read-through and interference. And of course, there's composition and load effects. So we haven't really quantified these things. Richard talked about the resource problems in an in vitro system. We have the same resource problems in vivo, but we haven't quantified them very well. How do we do that? And this is where the unintended consequences come from. And dealing with that in a multi-scale way where it's internal to the host, internal to the environment, and epidemic. So how do we make that work? How do we develop those theories? And I think that what you're seeing is people struggling with these things in the small, and I would argue we should struggle with these things in the large. And this is to gain credibility. We have to be trustworthy to our stakeholders to solve this problem, and trustworthy to each other, so we can work together more effectively. And standardized parts were a way of doing that, but is that the right approach? As engineers, I would argue that we're only 50% about the effect we produce. We're also about the trust we engender. That is, people have to trust you to get on an airplane despite what happened this week. People have to trust you to take your medicine. And they trust you not because what you built is positively, absolutely safe. It's because the risks are better understood and can be taken. The cost and benefits are true, so it's about getting things good enough. But that's where complexity comes from. A great deal of the complexity of an airplane isn't getting something to fly. It's getting it to fly reliably. And that's where the complex structures, I think, are going to come from. So I want to put this in front of our panel, who I'm going to invite up in a moment, which is to ask to sort of focus on questions like these, although I want questions from the audience as well, which are, what are your top 3 applications? Because without the applications, specifications and reliability, all that stuff doesn't matter. You got to get to the point where you need to build something. What are the roadblocks here technically and socially? How can engineering design approach these things? And how can we better learn from our basic biology colleagues for what the real issues are in making things operate reliably and robustly on evolutionary time and in space? How do we serve the public interest? And what is really going to transform us in 50 years? Not in 5, but in 50. So with that, I want to invite the panel up. I want to introduce 2 new members of the panel. The first is Jane Calvert from University of Edinburgh. She is a social scientist and has worked on everything from what is basic science and research to what is the sociology of life sciences, including genomics, systems biology, synthetic biology. And I think she had a great line. One of her synthetic biology colleagues called her a synthetic biologist-ologist. Second person is Sven Panke. He received his degree in biotechnology from the Technical University of Braunschweig. He's been through DSM. He's now at ETH Zurich and is a specialist in chemical engineering, pathway engineering, and high-throughput screening. So welcome the panel to the stage. Let's see. Guess we had not quite enough chairs. There we go. So you can't be accused of not having the right diversity here. We have people from an industry, people from basic biology, people from prokaryotes and from eukaryotes and multicellular organisms, sociologists and the like. So let's just start with the top applications. What are the things that economically and socially are going to have the biggest impact that should drive the requirements for a design, build, test cycle? Does anyone have an opinion about this? Either I get the question wrong, or it's a little bit embarrassing.
So,
I'll
start.
Yeah.
Now we're reconnected together. So actually, I think, Adam, you put some up there that I thought were pretty compelling ones. So you answered your own question.
Well.
But I'll give you three, and I think that they're not unrelated to ones you put up, but for me, they're the ones that I think about. So one is kind of a class of applications, but I think it's an interesting one, which is just looking at event detectors, right? So I gave you an example of one in my talk, but you can think about, how is it that we want to detect what's going on inside of a cell, or detect what's going on in the environment, or detect what's going on in our bodies, and that might involve trying to understand a sequence of events. These events might be kind of primitives. Is something bigger than something else? Did something occur or not occur? But we want to think about potentially a complex sequence of those and recognizing when that's occurred. And I think that requires more complexity than just detecting that A is greater than B. Needs some modularity and other things. And maybe, to Adam's point that he made earlier, often the difficulty when you look at other engineered systems that are complex, as Adam said, it's not about getting the fundamental basic operation going, right? The Wright brothers had something that could fly, but it couldn't fly, right, in windy conditions. It couldn't fly, when it was cloudy. There were a lot of things, and so it's about putting all of the stuff around it that makes it operate robustly, right? So I think that they become much more complex, not because we can't build a simple event detector that detects some sequence, but doing that in a way in which it works at multiple temperatures, it works in different organisms, it works in somebody else's hands. All of those things will generate complex circuits that have all sorts of compensation mechanisms built into them in different ways. So that's one. I think another one is to think about directions in which we build what are effectively artificial cells. So taking a little bit off of some of the stuff that Jason described, for example, can we build something that's completely artificial where we incorporate the metabolic pathways that are required to create energy inside some artificial cell and do sensing and locomotion and other sorts of things. We can do that for reasons of going and trying to do environmental remediation, find areas where certain chemicals are present and release them. So you got to get there, you got to sense it, you've got to release enzymes and other things. But now in a way that's completely artificial, so that in some sense this isn't alive, it's simply a nano device that we build using what biology has shown us can be done. So there's a second one. And I think thinking about artificial plants in a similar way, that is multicellular organisms, that again may or may not be alive in the sense that they can reproduce, but can do some of the things, can grow a material with integrated structure within it, for example. How would we do that, right? And I think those are three for me that kind of motivate.
I really like the idea of ubiquitous idiot lights. Where you have many cells out there whose sole job is to report to us on the health of whatever.
Yeah.
Health of the water, the land, yourself, whatever. And they're easily collectible and diagnosable for an event of interest to us.
Right. So put out a billion of these, each one doing different events.
Exactly, and just having that ubiquitous sensing out there. Of course, it has some scary repercussions, right? Anyone else have a,
I'm afraid I'm going to criticise the question.
Yes.
I should first say I'm one of the odd people you'll find throughout the conference who are not synthetic biologists but are kind of other people, so look out for us throughout the conference. I think that rather than saying what we think the most impactful things or what we as a synthetic biology community think are the most impactful things, maybe we should ask people what they want, partially because predictions in technology are notoriously hard. I mean, genetic fingerprinting, nobody imagined that was going to be used for forensic when it was developed. So maybe we should be asking industry, we should maybe be asking farmers in terms of their nitrogen fixation, we should be asking patients and other stakeholders, rather than us just without a wider discussion saying what the most impactful things are going to be.
I'm going to criticize you back. What makes you think we haven't asked them?
Sorry?
What makes you think we haven't asked?
Well, I mean, who's in the room here? So, I mean, I think we could do with having more people actually as part of the discussion here. And I know people have asked, but it's an ongoing process. It's not just a one-off.
Oh, and I agree with you. I mean, I think it's very important to have that discussion, which is why I cited a forum that isn't us. The World Economic Forum wasn't us; it was someone else.
Yeah.
But I do think that that's a very good point. And there's also a point of would you use this if we made it? So Steve Jobs is famous for saying, we have to make them want it, more or less. I forget what the exact quote was, but it was along the lines of they don't know what they want until we gave it to them. And there's a balance between these two things.
Yeah,
but I agree with you.
Yeah, I mean, if we're designing for users, we have to think about
the users
of these technologies. And also those World Economic Forum points you put up, they are not just technical challenges, they're social, political, and economic challenges. They're all wrapped into one.
This gets something very, I think is very important, which is the systems engineering side of things. A real system engineer will think about that as well, the economic pipeline, the social pipeline, and we haven't built models that capture this. JBay built a techno-economic model in part to drive how they design biofuels. And I think if you expand that a bit further for other things, that would be a hugely impactful thing to be doing. And as an application that people could get involved with, because you can play with it online, on the computer.
Yeah, extend the boundaries of what engineering is to encompass the social.
Yeah, I think it's a great point. Great point. I want to amplify something that Chris does. He's talking about nitrogen fertilizers as an issue. This is a huge issue, nitrogen fertilizers. So is phosphate. If you can get both these fertilizers out of chemical amendment and into things that allow you to fix it from the environment, it changes a lot of things. It changes a lot. It changes pollution issues. It changes some economic and security issues. It changes some social issues. And the question is how far could we drive that, and what are the impacts of eliminating fertilizer from a social and economic perspective?
Well, maybe even to take a little half step back. Yeah. When you mention all those things, nitrogen fixation, phosphate utilization, these are all part of an extraordinary number of microbial processes that are all exist in the sequence databases already. So Adam mentioned that there are 1 million organisms for which we have sequences. We must be approaching 1 billion genes now. And many of the most interesting functions, whether they be utilization, like you're mentioning, or chemical pathways or materials, are encoded between 5 to 100 genes and so on. So I think a big part of where SynBio finds its application is going to be how we can go in and the vast majority of those 1 million organisms are not manipulatable, and how we use the tools from SynBio broadly to access the information that's there and convert it into a form that we can actually move it into an organism of interest and gain access. I think it'll first be natural products and sort of reinvigorating pharmaceutical discovery, and then things like materials will come next, and ultimately all the amazing things that if you're a non-microbiologist, you're just constantly amazed by what they're able to do.
I think this brings up the second question. We can move on to that then, which is what are the technical and scientific roadblocks to getting things done? He mentioned the idea that you have to get things from this database of sequence into functionally well annotated, characterized things that you could also move around and transport between different organisms as a serious technological challenge we have right now. And I'd characterize as characterization and the taming of the wild as two major issues. What else do we have along those lines?
Well, I think one of the key things that we lack are really good understandings as to what species barriers are, from the genetics through to the very practical. If you're trying to put something in the membrane, how much does it matter if that membrane is made up with different lipids, all the way down to sort of the parts characterization and so on. So I think a big part of where we need to go next is expanding into several key organisms to try to see if we can build those out as platforms that enable the engineering. Yeah.
So something I think is missing, and not quite sure how to articulate it, but I'll try, is that in many other domains, I think what you see is a kind of well understood hierarchy of thinking about the problem from low level to high level that allows people to work at one level without having to worry about the details of the level below it or above it. And this is tremendously simplifying. So my background is in control theory, and most of the time I don't have to worry in detail about what the turbulence over a wing looks like and how that's affected by the details of the shape in order to design the control system that goes on that would allow the airplane to fly around. And it seems to me that in synthetic biology we're not at that stage yet. That is that many of us are having to work across all of those levels at once. We're having to, in some sense, design the application that we're interested in at the same time as laying out the DNA. Because we don't yet know, what's a subsystem and how do I represent that subsystem in a way they don't have to worry about it anymore. I mean, I know what the subsystem does. I have a description of that subsystem that allows me to reason at that level of abstraction. This is not just about parts, right? I mean, it's actually some mathematical level of abstraction. Yeah. And so I think that's sort of missing, and I think that's a challenge. Yeah. Right?
There's sort of three things here, and Lotta was commenting on this in her talk as well. This idea of encapsulation, that you can encapsulate a function, and then it's just an input-output structure,
right.
And then you can operate it with it as a drop-in object. It's something that we powerfully talk about, but rarely able to accomplish.
Right.
And so Chris wanting to move things around different organisms, figuring out what the species barriers are in part understanding those interfaces. In Laz's case, there are issues of where's the boundary? You want to comment on how hard it is to sort of figure out where to stop your study of the system, or what it means to be a module, since you put it in quotations in your talk?
Well, so I think what we have realized in the work that we have been doing is really that, so we started out very naively thinking that it should be easy to put proteins in certain places within a cell. And what we have learned is that this is tremendously complex. So really, I think that, and much more complex than we ever imagined. So I think that actually a major roadblock to trying to spatially organize proteins within a cell, but also organizing between cells, that's lack of fundamental biological knowledge. Again and again, we're being surprised about the complexities of the systems that we're working with. So I think it's very important to make the coupling between research in basic biology, basic biological mechanisms, and then synthetic biology, to bridge that gap so that we can really, in synthetic biology, capitalize on all the new insights that are coming from basic research.
That's actually, sort of our question 4 here. There's 3, but it's 4. How do we basically rely on our biology colleagues to teach us what's important about our designs, and also to sort of, I think define these interfaces, define the context in which they function. And actually, I'm quite impressed, Iris, you're able to get these complicated recombinases to work at all in these organisms. But if you think what kind of things that you as a biologist think that we need to know, what would they be? What are the things that you think or the things that you would want from us that would be, Jason talked about colors, and you said, no, I need resolution. I need resolution of microscopy and better imaging tools. What are the things that would be useful for us to build for you, and what things should we learn from you?
I think I would agree with Lotte that what we can give is the understanding of the biological system, and we are really very much at the beginning, because even the most simple question, why a hair points in this way, not in the other way, we don't know yet. But on the other hand, I think we can actually gain a lot from synthetic biologists, because, for example, when I see how fast DNA can be synthesized, and what we want to do is to kind of go into a genetic system, and we just do it like very old-fashioned way and slow, and students and postdocs are working hours and hours to get this done. And then I see, well, you can get it done in a day, and it's like, oh my God. So it's something like this, that this maybe, if it finds its way as technology, and also maybe because funds are obviously also an issue in our world. So it would make things really different how we can do things.
So Jason, going back to some of the physical issues, you've worked on the ribosome as a molecular engineering platform. And one of the things that's been a challenge since the last 30 years has been molecular engineering, this biomolecular engineering. You've benefited from there being ribosomal structures and a great deal of functional studies on these things. What's the status of this as you go slightly away from the ribosome? You think about the message itself, or you think about the tRNAses and the synthases and things like that. So what are the challenges that remain to be fixed here?
I think one of the challenges in this particular area and in the field in general is, really understanding the limits of how things scale and transplant, both in terms of, we know that subsystems are not infinitely specific, but how specific do they need to be for you to be able to use them in a scalable way, for them just to not catastrophically break? And in biology, I don't think we understand really how to do that.
How much crosstalk, or how much parasitism can we actually?
How much crosstalk can we tolerate in any given system, and when does it matter and when does it not matter? And what are the things that we would want to measure, or what are the things that we would want to know to allow us to predict that?
Yeah, so it gets back to some of the systems biology side of things, where you say, well, how do I measure, all the parasitic interactions that occur in a heterologous circuit in its environment? or for that matter, for any transplanted circuit, any other transcription factor, and in fact, it's promiscuous to some degree.
And are they parasitic?
And parasitic?
I mean, in the sense that, right, I mean, they may be there for a reason. They're good things, right? That, crosstalk is useful. Maybe it provides robustness, right? When we get rid of it, it breaks the robustness, right? Exactly. So now we need to design with crosstalk, whereas engineers were usually thinking about crosstalk as being something bad. Maybe it's something essential.
Right, so this gets back to some of the issues we have with how to deploy high throughput technologies or more directly to get this accomplished. And Sven, you've worked on high throughput screening, where you're trying to discover things as opposed to the screening process. But what are the challenges of making that work for different sorts of technologies, even for metabolic engineering? What have been the challenges of using high throughput screening to get things accomplished?
Any high throughput screen is only as good as the any high throughput screen is only as good as the library you put in. So if you have DNA synthesis capacities as they are emerging right now, then you can structure libraries in a much smarter way than you used to. And I think the output that you will get will be a lot better. The other point is, of course, that you need to get more sophisticated assays, more complicated assays, more complicated phenotypes to assay, and this will require novel assay formats, in particular with microtechnological tools, microfluidic tools, nanotechnological tools, stuff like this. So I think there is a lot of stuff to do, but I think that the technologies are kind of visible, so I think
you're okay with it.
I think we're doing okay. Yeah.
I think where we struggle with it is when we have to do things that are beyond the bioreactor, which aren't chemical synthesis. If I have to deploy something into a human being, for instance, how do I test it? And usually it's in a human being, which seems scary for some of our applications, especially if we're trying to build complex ones. So this gets to another question, which is how can we ensure the next generation of synthetic biology best serves public interest? This is a two-part question, one in which I want concrete things that are currently barriers to us going out into the world more precisely, but also just how do we engage? I think Jane brought up one, which is to ensure all the time that we're in conversation with our stakeholders, which is the people we're trying to serve. We are, after all, servants in that sense. So how do we engage that crowd well and effectively for both sides? But what are the things that currently prevent us from making progress, I think, in ways that we could address? Laws, regulations, economic issues, technological barriers that could allow us to overcome some of this communication barrier that we have.
Yeah, I mean, I think the question about the public interest, the tagline of the BioBrick Foundation is biotechnology in the public interest, and this is a really laudable aim. But then again, we do not know what the public interest is just in isolation. We have to find out what it is. And you mentioned earlier about risks being necessary to a kind of control of risks being necessary to ensure trust, when actually lots of work which has engaged the public shows that it's not necessarily primarily about risks. People aren't necessarily most concerned about the risks of synthetic biology. They may be more concerned about the purposes and the motivations, what's driving it, what's behind it, what's the why. So I think there's often a kind of looking at risk as if it's going to answer all the questions, when actually, when we're dealing with these uncertain systems, people, if they can see the why, then that's the most convincing thing. So
I think I want to emphasize, because I think that when I talk about risk, I often assume that we're talking about a strong benefit. That is, risk without benefit doesn't mean anything.
But who decides what a risk is and who decides what a benefit is? A benefit for one person may not be for another. If you talk about life extension, this may be wonderful for some people, but it may just be overpopulation and destruction of the planet for other people. So we cannot say what a risk is or what a benefit is. We cannot decide. We have to see what it means to different people in different contexts.
But don't you think, I mean, I'm going to challenge you a little bit here. This is absolutely true. But every technology that we introduce, in fact, every law we introduce, every book we write, has unintended consequences and impact. I can mention books, Mein Kampf, things like this, which are deadly in a sense, right? So the question is, what's different about our field?
I don't think there's anything different about it at all.
Right, so, well, I don't think it is. But I think where we are different is that because it is a scientific discipline, our ability to do actuarial calculations is better.
But it may not be about you can't do actuarial calculations about your purposes. about why you want to do it.
No,
That is a different issue. So sometimes the conversation is going along at two different levels and people aren't talking to each other. So
I agree, but I want to solve at least one level, which is to get policy to be placed on quantitative grounds.
Maybe policy can't be placed on quantitative grounds.
Not entirely, but we need to solve this piece. To have data-driven discussions. Now, I agree there's a whole other realm and an important and critical realm of discussion.
But these are data-driven as well. We had a public dialogue in the UK which drew attention to these questions of purposes, so they're not data-free.
Well, yes, sorry, yes, I agree. I meant from the biology side, yes.
I would like to add something because I feel there's a very easy way, and that is that we're actually absolutely honest about how we talk to people.
Yeah.
Because, for example, like hearing on the news or project, Craig Venter, I synthesized live. I mean, that makes people really uncomfortable. But what did he do? He put a piece of synthesized DNA in a cell. And I think it's just that we use what we say, what we're doing, and we're not exaggerating and using buzzwords and stuff. It's the same when people start to talk about human engineering or using humans as an experimental model. It's terrible because the thing is, even if you say what you precisely do, then people will be okay with it. Yeah. And I think we just need to say in the little stuff what we're doing and not overselling. I
think this very much connects to what Jane was talking about as well, which is it's the discourse that is about our story that we tell each other. And we're human. We like to tell each other stories all the time. We're also, I would argue, in a bit of an economic bind there as well, in that we tend to want to distinguish one from the other. There's 700 of us in this room. How do I distinguish one from the other of us? And it's one who tells a better story often. And I understand that this is not the ideal. And it's a challenge. I'm calling out the challenge of speaking plainly, of speaking plainly with truth. Is often boring. and getting back to Drew's statement that the field is becoming boring, maybe, and he said this himself, this may be a good thing, right? That means that we're getting over the hype. But there's going to be this balance between the two because marketing is important because it brings buy-in and it brings people together. And how do we balance these things? I think it's a challenge. Okay, we're nearly the end of time, 5 minutes. I'm sort of debating. Should we go on to the dream big? I think not. Is there anyone from the audience who has a question for our panel? If not, we can dream big. We do have one. Fifth question. Dream big. Sorry, not getting away with it. Dream big, guys. I don't know how you guys. When I was, I still do it. I still read science fiction. I'm sorry, and I love seeing things that were talked about in 1950 that are now true, and also seeing those things which are not. Flying cars, not really true. We can't predict technology. We can't. But I do think that learning how to dream about things that are realistic but yet very futuristic are very important. I think that we can extend something that Richard said about sensors. And being able to, we actually had this meeting at UCSF, I mentioned, we talked about the smart toilet, where everything that went in was analyzed. And imagine this toilet telling you, the status of nearly everything going on in your body based on biological readout. And so you get up in the morning and it tells you today you just need slightly more carbohydrate in your diet. Have some candy, it's okay. I'd like that. Back off on your ibuprofen or whatever is the new anti-inflammatory. And by the way, you're in a bad mood, so watch it. Things like that I think are going to will probably become more commonplace just because our ability to, our current desire in any case for a huge amount of information seems insatiable. And the ability to produce these sorts of ecologies in our body is becoming better. And so wouldn't it be great to have that ubiquitous sensor of yourself? What else? Yes, Sven.
So I'd like to open up a kind of alternative vision, which is that provided that we at some point solve the safety issue about genetic exchange, genetic pollution, however you call it, that we're on the way to create the possibilities for garage biotechnology, because if we at some point manage to really turn biotechnology into an engineering discipline, the side effect will be that it will become much easier. So think that the things that Chris now does with the help of half of MIT, I don't know, 15 years from now most probably will be able, hopefully, to do at the garage level. And if that's true, then I think our complete set of predictions that we might make now or not is completely irrelevant, because then somebody else is going to drive the innovation thing, because it has become much smaller. People, the route to idea and innovation has become much shorter, and it will be much more like the IT revolution went, which completely unpredictable outcomes. So I think I made a promise to myself that I'm not going to say anything about how it's going to look like in 50 years. But I think that's one of the reasons, because I think we're trying to work on a technology that would allow that the innovation process is basically taken away from us.
You know what I like about, I love the idea that this is what we want to have happen in a lot of ways, partly because I think that personalization is going to become a big deal. Again, I'm predicting, which is a bad idea, but personalization. So imagine that Pat Brown, who quit Stanford to start a company on food, on synthetic food, is successful. Now we learn from our ubiquitous biosensors that we need a certain type of food product for the next couple of weeks. You're able to synthesize yeasts that make that for you at home immediately. Now, that's a projection that Sven would never want me to make, because it's not going to happen. But I like the idea of this diversified, this ability to safely engineer for yourself what you need biologically as a dream. Jane, please.
Whenever I come to a conference, I always think a cure for jet lag and some kind of circadian rhythm thing would be great. And I've only come from Scotland, so I'm not suffering as badly as most people. But I suppose, again, I'm going to return to the issues. Who gets to imagine the future of synthetic biology? Who gets to write their sci-fi stories? And who has the discussion? I think that's really important. And if synthetic biology in the future still has kind of non-synthetic biologists like me hanging around in it, I'd be very happy. So it would be more about what is the synthetic biology community going to look like 50 years in the future, and if it remains open, inclusive, open to discussion, then I think that would be good.
So if Sven's vision wins, then you will be involved, right? Yeah. Because it'll be in your backyard. Excellent. Anyone else want to take a crack, close the session?
I have a comment, actually.
Yeah, Jason, please.
I mean, I think one of the things, looking at the sort of the first part about economic and social applications, I mean, I think one of the questions, I don't want to overemphasize this, but I do think that one of the questions that as a field synthetic biology should be looking at is, how can we get the understanding that we need of biology to make synthetic biology better? Because I think actually, we have to ask the question, do we know enough, right?
Right.
And if we don't know enough, then actually what we should be doing, at least part of the time, is figuring out what it is we need to know.
Perfect. Matches very well with Iris. Well, with that, I want you to thank our panelists for a wonderful morning.